11 years ago, Elise Lefkowitz and her family had the brilliant idea to bring together a fabulous group of women and honor her mother, Estelle Gelman, who bravely battled Alzheimer's disease until her passing in 2009. Estelle was the beloved matriarch of the Gelman family, and her elegance, grace, wit, dedication to philanthropy, and love of her family continues to be a guidepost for the generations of women following in her footsteps.
I remember my grandmother perfectly, from her dressing up every single day when she was healthy and going out and doing philanthropic events. She is and always will be the head of our family as far as I'm concerned.
She was the most elegant person that I knew. And I've always said, when I pass away, I want a legacy like Estelle. She would do anything for anybody.
For many, many years, she did a luncheon fashion show for the American Cancer Society because my father passed away from colon cancer. And every year it was a different designer, and it kept growing and growing and growing. I think it almost reached 1,000 people at one point. After she passed away, I thought I would really like to do something of the same sort. We've been lucky enough that many designers have wanted to be involved with our luncheon.
For the past decade, the Great Ladies Luncheon has provided a one-of-a-kind venue to combine high fashion and design and cutting-edge science, as well as the opportunity to celebrate many extraordinary women, including former First Lady Nancy Reagan, Coach Pat Summitt, Sharon Percy Rockefeller, and this year, former Supreme Court Justice Sandra Day O'Connor.
We decided to do a design at home event this year since we couldn't do a fashion show online. And another reason was because we were honoring Thomas Pheasant, who's been one of our biggest supporters since day one. He's one of the top designers in the country as well internationally.
I want to take this opportunity to thank and honor Elise. Your mother was very much like Esty in the fact that obviously she loved fashion and beauty and entertaining. You did an extraordinary job, and bringing this luncheon together and doing it in this way is super powerful, so thank you.
Elise, what you've done for the ADDF and for the Alzheimer's community is amazing. You are a great lady.
Cheers to the great ladies who were part of this event this year.
I'm toasting with red wine for all the great purposes it has for the brain. Cheers to the Great Ladies Lunch. Thank you for all you're doing for ADDF.
I think a great lady never gives up. They always strive to give more. And my grandmother strived to give more in every aspect of her life. Continuing on her legacy is critical to finding a cure for this horrible disease.
I think we have made a great impact, and I hope that this luncheon will continue on until the day we have a drug that stops Alzheimer's.
Hello, and welcome to the 11th Annual Great Ladies Symposium and virtual event. We're thrilled to have you with us this morning. I am Mark Roithmeier, CEO of the Alzheimer's Drug Discovery Foundation. In just a little while, you'll hear from some of our incredible scientists in a symposium sponsored by our great friends from Eli Lilly. For those of you who have not contributed to the event yet, we hope you will consider donating by texting ADDF to 243725. We are thrilled to share a special Justice Sandra Day O'Connor with the Great Ladies Award for her leadership and advocacy of Alzheimer's research.
Justice O'Connor was the first woman to serve on the United States Supreme Court.
But not the last.
Not the last.
It's
commonplace now to have women in government, but when 1981, when she came on the court, she was still unusual. This is before Sally Ride was an astronaut or Madeleine Albright was Secretary of State. She wasn't the Chief Justice, but she was the center of that court. It was often called the O'Connor Court because of her personal power and her ability to compromise and get along with sometimes difficult people. She could be tough. All through her life, she was challenged because she was a woman in a man's world as she was growing up, but she could also be warm.
Sandra was as close as I came to having a big sister. Justice O'Connor was the most welcoming, gave me some very good advice, not only when I was a new justice, but during my first cancer bout, because Justice O'Connor had breast cancer, and she was on the bench 9 days after her cancer surgery. Wow. So she was very clear about what I had to do. She said, Ruth, you have your chemotherapy on a Friday. That way you'll get over it during the weekend. You can be back.
She organized social activities for the law clerks. She really did give us advice about the importance of maintaining a balance in our lives.
One of Justice O'Connor's achievements on the court was she founded an exercise group.
Yes, indeed.
And she liked to have women clerks come to the exercise group. Yeah. And I failed to come to the exercise group.
I noticed.
She's always been dazzling at everything she endeavored to do, starting from being a student in primary and secondary school where she skipped grades and ended up starting college at the age of 16, finished her undergraduate work in 3 years, and 3 more years she'd finished law school.
Justice O'Connor was incredibly busy, but she always made time for her family. She had 3 sons, strapping boys who did very well in life and who loved her, a very close family. She was particularly close to her husband, John. Long, wonderful marriage. John was a traditional male, big-time lawyer in Phoenix. They were a team, a kind of a power couple in Phoenix. And then when she got to Washington, though, it was hard for him. He got a job as a lawyer, but it wasn't the same. And then in the mid-1990s, he started showing the early signs of Alzheimer's. And she knew what a struggle that was for him and for her and for the whole family. She took care of him.
Mom, when she rose to the court, felt that it was her obligation to perform at an incredibly high level, and she owed it to the country to do her best as the first woman on the court. She had a tremendous sense of obligation and guilt to take care of Dad, but she couldn't do both, and it was crushing her.
Research shows that the primary caregiver's health is dramatically affected, and life is shortened just from the stress and the realities of what it takes to look after someone with Alzheimer's.
And it really took a whole family intervention with all of us to finally get Mom to accept that
When she left the Supreme Court, she knew that she had, as she put it, 5 years to be relevant. And she get very involved in civic engagements of all kinds. She spoke about Alzheimer's. She testified about it. She spoke to Congress about the disease.
And I think that our nation is certainly serious about this deadly disease. I think we have to expand the research efforts, and we have to encourage the sharing of research data across the country with those who can help further this process.
Leonard Lauder reached out to her and asked her to become involved in the Alzheimer's Discovery Foundation. And he had her become honorary chairman of the board. She was very pleased to play a role in ADDF and to support its cause.
And then when she herself started experiencing the impact of Alzheimer's, she tried so hard to be able to continue her public work that meant so much to her. And because it meant so much, I always thought it took a great deal of courage for her to be willing to withdraw from public life and to make the public statement she did.
She was inspired by the example of President Reagan, who wrote a letter to the country, and so did Justice O'Connor.
She did it knowing that that would help people understand that Alzheimer's can impact anyone. And it gave her one more opportunity to be an advocate for continuing research and treatment options and so forth for Alzheimer's. And it was, I think, a sense of final act of leadership, public leadership.
She's still alive. She's in a care facility, but she kept going with her life as long as she possibly could, as vigorously as she could. And she's still a cheerful, warm person who's 90 years old and still trucking.
As incredible as the great lady O'Connor and her family. Years ago, Justice O'Connor, you came to us, and today you still serve as the honorary chair of our board. Well, the honor is all ADDF's.
Justice O'Connor has received a lot of awards, but the Great Ladies Award is a great honor to her. She deserves it. She is a great lady herself.
I just can't think of anybody who better fits the definition of a great lady. You have given us all so much, and you have done so much for the cause of Alzheimer's research and treatment. And to be recognized today is something you richly deserve, and I congratulate you.
I have long admired Sandra Day O'Connor as a jurist, an advocate for civic education and historic preservation, and a remarkable pathbreaker and role model for women and for men. So let us celebrate her and her family today for all that she means to all of us and all that she means to history.
If Mom were able to accept this honor in person, I'm sure she would express her appreciation to ADDF. I think it would be really important to her because it was an award from a group in which she personally invested of herself, her time and her energies.
My mom, the whole family is so impressed and grateful for the work that the ADDF does in promoting solutions for Alzheimer's disease. Nothing's more important to this country medically than coming up with a long-term solution for this disease. It's organizations like ADDF that can help play that role in a private capacity to find the most promising avenues for research and to help get funding into the right places and the right drugs at the right time. So on behalf of my mom, we gratefully accept the award. Thank you.
Hello, everybody. I'm Andrea Mitchell, and I am so pleased to be with you today. This is the 11th year that I've had the pleasure of joining many of you for this event, and it's my hope that we will all be together in person in 2022. The Alzheimer's Drug Discovery Foundation and I are pleased to welcome you to our signature scientific symposium featuring ADDF Founding Executive Director and Chief Science Officer Dr. Howard Fillett, and two of the ADDF's outstanding researchers, Dr. Frank Longo of Pharmatrophics and Stanford University, and Kent Leslie of Amylyx Pharmaceutical. We also would like to thank Eli Lilly and Company for sponsoring today's symposium. Today's panel will highlight two promising phase 2 trials exploring novel targets, both of which are funded by the ADDF and are expected to report new data in the coming months.
I'm Dr. Frank Longo. I'm the chair of the Department of Neurology here at Stanford University. We have been working on completing our phase 2A trial of our lead drug, known by its short name as C31, in mild to moderate Alzheimer's subjects. It's the first time the drug is in patients with Alzheimer's, and it's only a relatively small to medium trial, so it gives us some indications about its effects in Alzheimer's. And it gives us the information to allow us to design and carry out a larger trial that can give us a definitive answer, hopefully, if the drug's effective or not. Working with Dr. Fillett has been a real pleasure on so many levels. When we started, the common approach, and it has been for much of the field, has been to go after the amyloid. And I think amyloid's an important part of Alzheimer's. But our idea was to go much deeper than that and really tackle mechanisms that are allowing the neurons to degenerate and the loss of synapses in spite of amyloid being there. And so, going after a target that's not amyloid is different. It was especially different when we started. And I think what I really enjoyed about Howard was his broad perspective. He was one of the first to recognize that we would need to go after targets in addition to amyloid. And that perspective was very helpful for us. And now, years later, of course, the field broadly accepts that we will need to go after additional targets in addition to amyloid. It really dawned on us that if we could invent a drug that controls the mechanisms that dictate whether a neuron stays alive and whether it maintains its connection, that in fact might counteract the degenerative mechanisms that our people were discovering occur in Alzheimer's disease.
My name's Kent Leslie. I'm the Chief Scientific Officer at Amylyx Pharmaceuticals. And here at Amylyx, we're developing a combination therapy, AMX0035, for the treatment of neurodegenerative diseases, focused at this time on ALS, amyotrophic lateral sclerosis, and Alzheimer's disease. Right now, Amylyx is running a Alzheimer's disease clinical trial. Our hope is at the end of this, we get a really good idea for what the drug is doing and whether or not what it's doing is targeting processes that are relevant to Alzheimer's disease and its progression. We expect these compounds to be slowing neuronal death processes, targeting multiple causal pathways simultaneously should allow us to target more of the features that are really driving neuronal death. AMX35 is the first combination therapy in clinical trials for the treatment of Alzheimer's disease. Hopefully not the last, but I do think we are among the first. But in neurodegenerative diseases, it's not often the case that you find a single genetic cause or a single culprit that's associated with the disease. There's quite a few things going wrong, and if we're going to target those, I think we're going to need to really target all of them at the same time. I think the more we understand about really what's going wrong in Alzheimer's and other different types of dementias, I think that will directly point us to sort of paths of better understanding cognition, better understanding language, better understanding speech, better understanding of executive function. We would not have been able to run our clinical trial without the ADDF. I view the ADDF as sort of this ubiquitous presence in the Alzheimer's landscape. It's sort of tough to imagine Alzheimer's without the ADDF. But I think our company and our story really shows the niche that the ADDF fills, an incredibly important and sort of unique niche within the landscape.
Hi everybody, I'm Howard Phillott. I'm the founding executive director and the chief science officer for the Alzheimer's Drug Discovery Foundation, and it's my privilege to be here with you at our Great Ladies Luncheon. Frank, your current drug in phase 2 is called C31. I was wondering if you could tell us how C31 actually works.
This drug is what we call a small molecule, which means it can be taken as an oral capsule or pill by mouth, and it gets into the brain. And what it does is it modulates the activity of what we call a receptor on the surface of cells, and in this case, a receptor on the surface of nerve cells. And by modulating that receptor, it counteracts the degenerative processes that are triggered by amyloid and pathologic forms of tau. And by counteracting that signaling, it makes the neurons resilient to degeneration.
I understand you're conducting a clinical phase 2 trial, which is a regulatory trial on the path to FDA approval. Can you tell us something about the design of the trial and when you expect to report some of the results?
Our phase 2 trial is actually a phase 2A trial. And what the A means is what we call an exploratory endpoint trial. No one's ever gone after this kind of mechanism before. So we're measuring a number of outcomes, and because it's the first time, we don't know yet which outcomes we would have the most effect on. Our plan is to report on the findings of this coming July at the Alzheimer's meeting. It's not what we call a formal efficacy trial, where in that kind of trial, one declares at the beginning what they think, what outcome they might address. And that's eventually the kind of trial that the FDA would need. So ours is a step before that.
Hi, Kent. Great to have you. Could you give us a little background on how you actually got interested in Alzheimer's disease and Amylyx, how that was founded?
Unfortunate. I don't have in my immediate family anyone affected by Alzheimer's disease, The interest for me in Alzheimer's actually came from, we'll say, sort of the basic neuroscience. AMX35 is a combination of two small molecules. So these two small molecules are repurposed compounds. So each of them have distinct medical purposes. So the idea is that in combination, targeting with one molecule excess misfolded proteins, and with the other molecule targeting mitochondrial dependent cell death signaling, the idea is that in combination they're each targeting processes involved in neurodegeneration, and in combination we expect them to be more active than the individual agents. We see a direct link between, is the neuron able to generate sort of an adequate amount of energy, and if not, what we see is both these cell death signaling processes from the mitochondria, as well as an inability for the cell to keep up with its protein folding demands. The most common sort of understanding of mitochondria are they're the powerhouse of the cell. What they do is they take glucose, they take oxygen, and what they produce are usable energy for the cell. And when that process goes awry for any number of reasons, neurons disproportionately are affected. I think we got very lucky, and these two compounds had a history in human patients, allowing us to speed up time to clinic. And we only evaluated these two compounds. We had an idea that these two might work, and interestingly, they did.
Kent's trial is a great example of another important emerging area. As our ability, to measure things in patients gets better and better, I think we'll find underlying patterns that contribute to the degeneration that might be amenable to targeting by drugs that we already have, or molecules that we already have out there, and they weren't developed for Alzheimer's, right? They may have been developed for something else, and that will really accelerate things, because then you have drugs that we know are safe in people. There's some experience in people. No one ever realized they might be relevant to Alzheimer's, but through that kind of research, now the program can jump right into testing those drugs in a much more accelerated way, and that's going to be great for the field.
But with these compounds, the FDA let us jump from sort of a conceptual idea into a phase 2 study in 100 patients without having to go through healthy volunteers, without having to go through these sort of phase 1 safety testing.
Frank, I recall when you were at the University of North Carolina, when you were chairman there, I think it was around 2007, if I'm not mistaken, that we talked about starting a new company. And of course, that's when Pharmatrophics was started. Why did you feel that starting a company was necessary around that time to accelerate the drug development efforts?
Great question. We never had as a plan of starting a company. I thought that if we could invent these drugs and show that they work in mice, that they'd be all set to go for a large pharma to do the next steps. And what we learned is that that was still at a very risky point in the project, and it would be difficult, for most pharma companies really to take up the project at that point. So we learned about the gap that exists between developing something in the university lab and getting it mature enough or de-risked enough that our colleagues in pharma could take it up. So how does one get across that gap? The answer was starting our own company and doing those in-between steps of work. And the challenge, of course, in starting a company like that is the funding. And so ADDF's biotech program really made it possible for us to get off the ground.
I think there's an accelerating ability to do better clinical trials, particularly in phase 2, to determine whether a drug is working in a much better way than we could ever do before. And for me, that's one of the most exciting things, that all this basic research and all the biomarker work has led to a time when I'm really excited about the way we're doing clinical trials, and I think the future is bright. Well, I want to thank everybody who's listening for your attention. I particularly want to thank Frank and Kent for working with ADDF and participating in this program today, but especially for their amazing work in developing new drugs for Alzheimer's disease. And we hope that in the next few months that we're going to hear amazing results that are very positive from your drugs, and we look forward to working with you further to help bring those to market and to patients who are suffering this nightmare. Hi, everybody. I'm Howard Fillett, and thank you again for joining us. And I particularly want to express my sincere thanks to the Gelman family for many years of working with ADDF, and to say also what an honor it is for us to be working with Sandra Day O'Connor and your family and everyone else that's on the phone that makes our work on the video conference that makes our work possible. And I really encourage you, no question is silly. Please ask questions. This is your opportunity to ask questions to me and Frank and Kent about what's going on. I'll start off. We've heard a lot about biomarkers. I'll start with you, Frank. What roles are biomarkers playing in your clinical trial?
Well, sure. Thank you, Howard. And I'd also like to add my appreciation for being at the event today and everyone who's attending and the team that put it together. You know, biomarkers are very important. Biomarkers allow us to know if a treatment's having an effect, and those include certain kinds of brain scans to see what's going on with degeneration in the brain, or measures of protein in the spinal fluid. Sometimes they include blood tests. And it's really the only way we know if we're affecting the degeneration that occurs with Alzheimer's.
Well, thanks, Frank. And could you mention other specific biomarkers that you're particularly looking at in the phase 2A study that you're hoping will give us a signal as to whether or not your drug works?
Yeah, so in our case, for our trial, you know, our goal is that our drug, significantly slows down degeneration, and we're giving it in fairly late patients that already have degeneration, mild to moderate. Our hope is that if our drugs given earlier would even delay or prevent the disease from starting. And so we're looking at certain parts of the brain that undergo atrophy or shrinkage in Alzheimer's disease. And so we're using very high-powered imaging to measure the shrinkage rate of those parts of the brain. And we're also using a certain kind of PET scan of the brain that tells us about the nerve cells and their connections and whether they're functioning or not. So those are probably our two most important measures.
Thanks, Frank. And Ken, could you tell us something about what role biomarkers are playing in your clinical trial?
Absolutely. Well, the biomarkers are the core of our clinical trial. So right now, patients in our study have been gracious enough to undergo 2 lumbar punctures at the beginning and the end of the trial, and multi-sequence MRI. So exactly as Frank was mentioning, certain regions of the brain, the hippocampus, particular cortices, tend to atrophy at a more rapid rate, and you can visualize that with a high-powered MRI scan. So really, one of the key outcomes for our study is looking at the rate of volumetric decline on a brain scan. But as Frank mentioned, looking in spinal fluid can be incredibly telling about whether or not our drug's doing what we hope it's doing. So in particular, biomarkers such as mentioned before, amyloid and tau can give us insight into, are these Alzheimer's patients? What do they look like at baseline? But we're looking into, as was discussed earlier, neuroinflammatory biomarkers, things that may indicate that the immune system is overactive, neurodegenerative markers, neurofilament, neurogranin, things like that. So the nice thing is these biomarkers really allow us to translate and see if what we're seeing in the mouse models and the cell lines is really what the drugs are doing in people. So biomarkers are absolutely critical to our clinical development.
No, thanks, Kent. Getting back to you, Frank, we have a question from the attendees. They want to know if C31 could be administered to people who are at risk of developing the disease. And I guess that plays into the role of biomarkers in prevention and what your vision of the future is in terms of how, if C31 is successful, it might be used as a preventative agent.
Sure, that's a great question. And the way C31 works, it prevents what we call the degenerative signaling mechanisms that occur even at very early stages of Alzheimer's, the stages where even there's no dementia yet in people. And so we're very excited about doing that kind of trial. Our dream, our first trial that we're just wrapping up now is in what we call mild to moderate stages. People already have the cognitive loss. And we're going there first just to see if we can move the needle in the right direction in these more advanced stages. Our dream is to give our drug before the dementia starts. So using biomarkers to identify people who are at risk, who we would predict that unfortunately will get a dementia in a few years. And we think that our drug would substantially delay or even prevent that. That's what the mouse versions of Alzheimer's are telling us. And of course, now we need to find out if that'll work in people, but we're optimistic.
So we have a question about using biomarkers in the clinic. The question is really, can someone be tested today for whether or not they have Alzheimer's or they're at risk for it, even if they don't have any symptoms? And I'd just like to take a crack at that by saying that biomarkers have been part of ADDF's mission from the very beginning, from our incorporation papers, which laid out the vision of ADDF from the Lauder family. And I just want to say that in terms of success, that ADDF has supported not just the first biomarker to come to the clinic, namely the PET amyloid scan, in which a person who's interested in knowing if they're at risk and even have Alzheimer's disease in their brain 20 years before they have symptoms can go to a radiology office today and get that test. And the answer is yes. The only problem with it right now is that it's not paid for by Medicare or any insurance company, and so it has to be paid for out of pocket. But we're very excited that if one of these anti-amyloid drugs comes to market, that test will be necessary to get those anti-amyloid drugs. But the second and more recent and probably even more relevant advance to the question about how can one get tested today for Alzheimer's disease, even if you don't have symptoms, is that there is a blood test on the market today from a company that ADDF has been investing in for several years now. The company's named C2N. It's spun out of Washington University, and it was approved by Medicare and is now approved in 47 states, unfortunately not New York, but in most other states, where I've actually been able to help people to get this blood test for Alzheimer's disease, where it really correlates with the PET scan and has a high degree of predictability in terms of asymptomatic people as to whether, along with some genetic testing for apolipoprotein E, a person is at risk or even has Alzheimer's disease in the preclinical phases. So just let me say on clinical aspects of biomarkers that ADDF contributed not to this, the first one, but the first two blood test and a PET scan that came to market for clinicians to use to make a diagnosis of Alzheimer's disease and to help people who want to know if they're cooking Alzheimer's disease. And so with that, let me move on to another question that we've had. Frank, you and Kent both chose to take a route of developing drugs through really early stage biotechs, which is a difficult path. Could you describe to us why it was advantageous? And I'll start with you, Frank, maybe again, and ask Kent to follow on. Why didn't you were at Stanford and you could have done it at Stanford, and our first investment with you was at Stanford, and even before that at North Carolina when you were at UNC back in the early 2000s. But we talked about a biotech, and how has that worked out, and what was the advantages of you starting a biotech and doing it outside the university?
Well, sure. So the university is a wonderful place to do the initial first basic steps of research. When we have new ideas, we can do some of the early testing with growing nerve cells in a dish. We can work with mice, but once we decide that we really want to move into people and clinical trials, the university, is not set up to do early stage clinical trials in all cases, unless it's an already established drug that a large pharma is working with. And really, one has to be nimble and flexible, and that's where a startup, small biotech, generally is the best model in terms of flexibility, creativity, flexibility on funding. So ADDF, we were too early where a large pharma would partner with us, too risky. The university was not quite the most efficient place at that stage of our work. We're right in the middle there. And our goal was to do the next step. And ADDF actually made it possible for us to start our company that's been successful in finishing 2 phase 1 trials for safety and just recently completed phase 2A trial. So it's actually worked out well.
Yeah, thanks, Frank. Frank, I think you were the first company that ADDF did a startup. We've done about 12 startups, and we have that through our biotechnology founders program. So thank you very much. Kent, maybe you can speak a little bit, not to the value of the conduct of the trials, but also how a biotech might be more helpful in getting the financing, which I know you've been successful at as an early stage company.
Yeah, absolutely. To add to Frank's comments, the one thing I'll add is there are quite a few skill sets involved in sort of human clinical testing getting to that stage that are really unique skill sets, whether this is toxicology, pharmacokinetics, process manufacturing, things like that. And scaling up from a mouse study to running a clinical trial at 10 centers around the United States is a really, really significant leap that I think universities could do it, but they're really not designed for that purpose. But in terms of the financing, at this point, we've raised money from grant through the Alzheimer's Association, the ADDF, and that funding really helped to catalyze funding from private sources, whether they're high net worth individuals, whether they're venture capital groups. And I think the structure, the sort of long-term vision, things like that, it's very nice to be able to bring in venture capital funding that understands the system, sort of understands what it takes to move into a phase 1 study, a phase 2 study. So I think in our experience, having an outside biotech company that can raise lots of different types of funding, whether it's grants, whether it's venture capital funding, really provides flexibility that I think is really critical to clinical development. So I hope that answers the question.
Yeah, thanks, Ken. Well, I see we're coming towards the end of our Q&A, and I guess to close, maybe I can ask you, Kent, you and Frank in line. You know, we've had so many failures in our field. We haven't had drug approval really since about 2003, and there's a lot of skepticism and nihilism out there in the investment community and among patients and their loved ones. You know, but I think among us and among the field, I have a sense of great excitement right now that we are going to be successful in the next few years. And certainly ADDF has seen a lot of results, positive results. In the phase 2A, we're currently funding 35 clinical trials, and we've had readouts on 5 last year, and we're looking at 6 more in the coming months. And they've all been really interesting, and we'll, you know, directly gain more capital. But Kent, maybe I could ask you about your vision and your point of view on where we are today and how excited you are. I know you're kind of biased because you're running a company, but maybe you could give us a perspective.
Yeah, I think you have to be eternally optimistic to run a company in the Alzheimer's space. But I think in the 6 years that I've been working at Amylyx, things have changed dramatically. I would say 2014, 2015 were really sort of the lowest point in terms of optimism. Quite a few anti-amyloid drugs had failed, and there wasn't really a clear direction for where to go next. Tau antibodies were too early stage, and really the breakthroughs on the biomarker side and on the genetic side hadn't yet materialized. At this point, there are so many different approaches in the clinic, whether it's targeting gingipains, targeting the neuroinflammatory system, targeting ApoE, so many different things in the pipeline that we're so much more diversified than we were 5 or 6 years ago. And I think we're really in a great position. So I think the next year or 2 are going to have more clinical advances than we've seen in the last 10. So yeah, I'm very optimistic.
We'll see what happens in June with the Lilly drug. There's a lot of excitement about that as potential really big breakthrough. Frank, do you want to comment on your perspective, like where we are right now?
Yeah, sure. Howard, I'm very optimistic for 2 major reasons. Number one, we are learning so much more about Alzheimer's and how it works. There are large studies out there now where we get human information about what's going on in the brain. So we really understand why the brain is degenerating so much better in the last 5 years. And then when we look at our therapies we're developing, we're very excited. In our case, it turns out we're targeting some of those major mechanisms that people are identifying. And then second, you know, which you mentioned the many trials that haven't worked out well in terms of finding a treatment effect. You know, we are, Kent and I and our other optimists, we don't call those failures, because with each trial, we learned a lot. The people in this field are great about sharing their data and sharing their experience. And we always learn from a well-run trial, regardless of the outcome. And then we use all of those learnings and make our trials better. So we're going to start seeing treatments that have some effects. I'm optimistic.
No, thanks, Frank. And I know I've been doing this over 40 years now, and I've never been more excited. And the advances, like, for example, having a blood test on the market for Alzheimer's is just hard to imagine, and we have one. And that's just one thing. And as I say, there's over 120 drugs in clinical development right now. More than half of them are not amyloid and not tau drugs. They're innovative mechanisms, as Frank said. And I do think that the next 5 years or so, and maybe even in the next few months, are going to be the most exciting times as we harvest all the 40 years of research that we've been doing in Alzheimer's disease and bring some of these drugs and some of these ideas to market for the help and prevention of Alzheimer's disease ultimately in human beings suffering. So I want to thank Frank and Kent for a great session and for being part of our event here. And again, thanks to all the donors on the line and all the contributors. And again, to the Gelman family and to the O'Connor family for joining us today. So thanks for your attention. We really appreciate it. And thanks for your support. Without your support, we would not be able to do our work.
Thank you.
And now it is my pleasure to introduce you to two outstanding women, Aerin and Jane Lauder, who carry on the legacy of their grandmother, Estee, and her sense of style, flair, elegance, beauty, fashion, and of course, philanthropy.
The thing that I always think about my grandmother and remember in the lessons she taught me, she was able to build this incredible business by her determination and her passion. And I remember when she would tell us about sitting outside of a buyer's office all day waiting, even if she didn't have an appointment, that perseverance and persistence. And so when I think about myself and I think about moments at work when you think, oh my gosh, I've gone to the end and I give up, I would think about Estee and then I would redouble my efforts. And the determination and passion that you have for your business is something that's critical to be successful.
I think the interesting thing about Estee, from my point of view, is she really was like the modern woman who loved lifestyle. You know, she loved home, she loved business, she loved family, she loved fashion. She was probably one of the first to create that sense of kind of fantasy and dream. She really taught me a lot about kind of going with your intuition and staying true to who you are. And of course, the one thing she always told Jane and I is have passion and love what you do. And, you know, we're very fortunate to love what we do and get up every day and be excited to start the day. It was always this attention to detail that really, I think, was very much a part of Jane and my life. I mean, we really grew up with this incredible style icon. And my mother has a wonderful sensibility as well. So we've learned really, I think, from the best.
You've done an incredible book around how you entertain, and now more than ever, especially during COVID, entertaining for your family is a great way to connect and be personal. So I'd love to know a little bit about your book and the idea behind it.
Estee had this wonderful idea that even if you're having a cup of tea, use a beautiful teacup. Don't save it for a special occasion. And that's very much the concept about the book. It's really about taking that time for people that you really care about.
As a family, we have so many great traditions that Estee started, and I think the first one I would say about entertaining is that she was very focused on making sure that there was always enough food. So I think maybe that's the way we think about it as a family. But I also think what she did, which I was always impressed by and which I've tried to do in my own entertaining, is giving gifts. And so she would give gifts of perfumes or cosmetics to people that would come and, dine. I think it's that wonderful moment of surprise. And I know Aerin does it even better, so I'll turn over to Aerin and ask her the same question.
She really had this incredible attention to detail, and whether it was the flowers or the monogrammed place card or the beautiful menu card or the little gift as Jane discussed, or even an unusual menu. She was known for these kind of wonderful, glamorous dinner parties and would be hosting the Duke and Duchess of Windsor and Princess Diana, and she would have this silver bowl with vanilla ice cream and she would pour champagne on top of it. And it was just such a moment, and her sense of style and kind of passion for life was really shown in the way she entertained. Philanthropy has always been a very important part of our family, obviously with the Breast Cancer Research Foundation that my aunt created and with ADDF that my uncle and my father did in 1998. The fact that it's 100% of the proceeds go to research and doctors, and it's just really extraordinary.
I think about my father and uncle starting this foundation. You think about the lessons that we all learned from Estee, and it goes back to the determination, the passion, never taking no for an answer. If you think about Alzheimer's, it's so many years of people trying to find a cure, trying to find ways to eradicate this disease. And I think the fact that they had a vision and they worked with all the best people in the industry, and the progress that's being made is extraordinary.
On behalf of the ADDF and the Lefkowitz family, we are pleased to present the second annual Estelle Gelman Award to internationally recognized interior designer and Alzheimer's advocate Thomas Pheasant. And now, longtime ADDF friend Andrea Mitchell takes us behind the scenes for a special visit with Thomas at its Georgetown Design Studio.
People want designers to be a style, like this person's a modernist, this person's a traditionalist. I think for me, it just evolved. It wasn't a plan. Using my foundation of classicism and creating simplicity in the work that I do, whether it's traditional or modern or a mix. It's kind of these root elements that I use for all my work that kind of come through regardless of what the project is. I'm never home. I'm always traveling. So having a year at home made me really appreciate the importance of my environment. This is kind of my pinup board. So this is 18 months, and it started with my trip to Japan, and I came back just totally taken with the culture and the beauty in every aspect of their life. So McGuire had come to me and said, "Tom, we're ready to do a new collection. Do you have any thoughts?" And I said, "I've got a lot of thoughts." These are some of the images that I took in Japan, and these are the first round of prototypes. Like we have the kimono chair, and using totally natural materials and natural woods and tones to kind of.
The tones and the textures, it just completely fit with what you have done in other classical genres.
It does. It's just it's just an extension, It's just this global extension, which is great.
Tom Pheasant, first of all, congratulations. Thank you for all you do for ADDF. I have been privileged to have known you, and you first as an architect trained. Mm-hmm. I think that fact really influences your design. Mm-hmm. So talk to me about your design aesthetic, how that evolved and how you're expressing it now.
At a young age, I remember the school field trips to the National Gallery, and I remember I was literally in 3rd grade. I walked into the National Gallery, and we had to be single file in the big rotunda, and I remember looking up and thinking, I want to live here. And for a kid who had never been in a place like that, who grew up in the suburbs of DC, that it was a major impression, and I can recall it literally. My focus is always on simplicity and creating open spaces, because I think in any city, the biggest luxury is space to breathe. I mean, people invest a lot of money in design, and I feel like it should have a life and a breath long after I leave the space. It should look effortless, but it takes a lot of work to create something that's fulfilling and simple. I say that, inspiration is a really important part of design. But inspiration isn't replication. It's about seeing something, experiencing something, a color, a space, a place, and then letting it just come inside you. And then once it's inside you, waiting for that chance to let it out. But when you let it out, it's new. You're not copying what you've seen. You've understood what you've seen.
Tell me about your connection to Alzheimer's.
Well, like many families, our family was hit twice. When I was in college, my grandfather was diagnosed with Alzheimer's. This is my mother's father. Many years later, my mother was diagnosed, and it was a whole new revelation on experience. And of course, being a mature person and being there, being one of the caregivers, and being experiencing and watching the whole process, it was it's such an emotional roller coaster. And there's fear in it, there's anger in it, there's beauty in it, believe it or not. There are moments that are just unbelievable where the person that you're losing kind of comes back, and then she goes away. And it's not only watching my mother, it's watching my father deal with it, my brother deal with it, the people that love us deal with it. And I was delighted to support ADDF because we've got to find a cure. We have to do something. It's not only for the people who are afflicted with the disease, it's for all the people that love them. Because of that personal connection and experiencing it, it really sort of promotes that need to help and to look after a bit for everybody and to participate. I'm thrilled that Elise and Mark have done such a great job in pulling the DC community together.
They have been amazing.
They are. They're amazing. And I mean, they're motivated through their same, family connection to the disease. And there's nothing more empowering than that, really.
That's why you being the winner of this year's award is just so meaningful to them and to the community.
Well, I'm honored to be honored, but I'm more honored to participate and to do what I can do. And if we could get everybody to invest in this and push this forward, I think we're going to find ourselves. I'm hopeful and optimistic that we're going to find ourselves, in a decade from now, in a whole new place. And that's the energy that's behind the organization.
Congratulations on that. Congratulations on the Estelle Gelman Award.
Oh, thank you.
And thank you for everything you're doing for ADDF.
Well, thank you to ADDF.
We should all be very grateful to the organization.
Yes, we should. Great.
Now, more than ever, we rely on your generosity to turn today's hope into tomorrow's scientific breakthroughs. We hope you will consider donating by texting ADDF to 243725. None of this would be possible without you, our generous donors. Truly, your philanthropy makes scientific breakthroughs possible. We are especially grateful to our outstanding founding executive chairs, Elise and Mark Lefkowitz, and their children, Charlie and Matthew. Thank you for everything you do. And of course, thank you to Leonard and Ronald Lauder, whose vision and forward thinking continues to be our true north. We wouldn't be able to do this without our wonderful co-chairs, Amy Baer, Lynn Blitzer, Kristen Ray Checky, Charlie Lefkowitz Crowley, Dana Landau, Kelly Ripken, and Lorraine Wallace. We also want to thank our honorary chairs, Andrea Mitchell and TJ Oshie, our core support underwriters, and our Ladies and Gentlemen's Committee. Lastly, we want to extend our gratitude to our contributors, especially our breakthrough, leadership, patron, and ambassador sponsors. A special shout out to everyone who appeared in today's event. We are so grateful for your support to our mission. And to everyone, please be safe, please be healthy. We thank you for so contributing to this cause, and we look forward to seeing you live in person for the 12th annual Great Ladies Luncheon next year.